Okay, I'm recording very early. So if you're watching this video, you can skip ahead until you see the camera moves. Obviously, the camera right now is showing the ceiling of Hamblin Auditorium. Um, so once you see the cameras pan and zoom towards the screen, then you'll know you should stop fast forwarding the video. But since you're here early, so to speak, the first attendance word, again, because you can just fast forward through this, but before you fast forward, say the first attendance word is rock, like Chris Rock or Dwayne The Rock Johnson or the movie The Rock or the genre of rock and roll. That's the first attendance word.
All of life stems from cells' ability to reproduce themselves because that's what allows organisms to develop and grow and heal and keep from dying for as long as possible. This particular kind of cell division is called.
You here, early cook?
I
Everything okay?
Can you hear me?
Yeah, can you hear me?
Yes, I can.
Okay,
I tend to show up to places early.
that's a
I'm
good
sorry.
thing. That's a
I'm
great
sorry
thing.
I couldn't be there today. I could not show up.
Hey, it's no problem at all. Really, honestly, it's easier to be here, because when you're online, you have to pay attention, close attention, closer attention. So, no need to apologize. It's all the same to me. Plus, today's, like I said in last week, or excuse me, earlier this week, I was saying that one of these days we're going to have to not use the auditorium, and I think it's next week, but I'm not. I still haven't confirmed that. So today they might show up and be like, "What are you guys doing here?" And then I might have to go to the auditorium. So it's good that you're online and not here. But anyway, for anybody, I've already started recording. So if anybody's watching this video, you can still keep fast forwarding. Like I said, it's that screen where it says my name. Once you see that screen move and go towards the projector, then you'll know to stop fast forwarding. But as I'm sitting here working, I am going to be playing some mitosis and meiosis videos to, see if I like them or not. So it is today. So today's the day you're using the. Yeah. There we go. Perfect. Like I was saying, today is the day. So Michael, I'm going to get offline. No, I'm just meeting with somebody really quickly. That's fine. You take your time. Okay, thanks. So yeah, Michael, I'm gonna move upstairs. So if anyone else shows up online, you can, if you don't mind, just tell them what's happening. I'm moving to the upstairs room.
All right, I'll tell them.
All right, I appreciate it. Okay, I'm back. Sorry about that. So I accidentally closed out the meeting too early, but it didn't matter because it looks like it's still recording and everything. So once again, I guess if you're watching this recording, you might think, oh, this is the time I should stop fast forwarding because there's been some big changes. And there have been, but you can still keep fast forwarding. I had to switch rooms. I guess once you see the PowerPoint presentation pop up on the screen, that's when you know that it's time to stop fast forwarding and start watching the video. Anyway, I don't know if you can hear me, Michael, but if you can, since you're here early, I'm gonna go ahead and give one of the first one of the first words. Oh, Caleb too. Awesome. So you guys get extra points. It's really early. So the first, or one of the first words for attendance, extra credit for Cook and Melton, will be paper, like a piece of paper. That's the first word or one of the first words for attendance. And for you two who are online currently, that is extra credit. So that's it. Anyway, I'm going to get back to work here, and I'll see you guys in a second, or in a few minutes. In the meantime, I'm going to start playing some mitosis videos and see if there's any good ones I'd like to share with you.
Chase, make more of what's yours.
If you're finally ready to take a trip without the kids, why stay at a hotel?
Have you ever looked down at your leg or your arm to find some cut and you have no idea where it came from or how you got it? So you put a bandage on it and a few days later it's gone. It's all healed and you don't even think anything of it, of this amazing process that causes this to happen. Or let's say you're looking at your nails and you notice they're a lot longer since the last time you cut them. Or let's say you're looking in the mirror and you notice that you are a lot bigger than you were when you were 5 years old. What do all of these things have in common? Well, one major thing they have in common is mitosis. Mitosis is a type of cell division done by most of your body cells, and it's really important for cells to divide. If they didn't divide, you wouldn't grow. I mean, how do you grow if you can't make more cells, right?
All very true. So far, I like it. It's a little bit childish, but what are you going to do?
So one reason why you're bigger than you were when you were 5 is mitosis. Mitosis also is great for repair of damage. If you have some kind of accident, like when we were talking about that cut on your arm or leg, well, you want to make sure it gets repaired well. So you have to make more cells to do that. Mitosis is great for that. Now, it's really important to understand what it is not. Mitosis is not a process that makes sperm or egg cells, because that's something different called meiosis, which sounds like mitosis, unfortunately, but it's a different process. Mitosis is done to produce body cells. Mitosis makes identical cells. That is the goal, identical cells. So if you're trying to make more skin cells to
That's a very important key too. They make identical cells. A lot of what this next exam, so this will be exam 3 that this material is on, a lot of it's going to be differentiating between the difference between mitosis, which is what this video is about, of one particular type of cellular reproduction, and then the other one is called meiosis. It's a different type of cellular reproduction, and so you're going to differentiate between the two of them a lot. So again, mitosis, like she just said, that makes a perfect copy of the cell.
replace worn out or damaged skin cells, you don't want to start suddenly making stomach cells there. That would be ridiculous. You want to make sure you have identical cells replacing what was lost. So mitosis makes identical cells. It's a really important thing. Now, it's also important to understand that your cells are not dividing all the time. If all they did was divide, it would just be rapid, crazy growth. In fact, this is kind of what cancer is. Cancer is uncontrolled cell growth. We have a clip on the cell cycle and what the cell is usually doing most of its daily life, which
I'll have to check that clip out because that sounds pretty good. That's another topic that will be in this chapter and on the third exam. We're definitely going to talk about cancer and how cellular reproduction rate relates to it.
is actually a phase called interphase, where it's growing and replicating its DNA and carrying out its daily cell functions. That's where cells spend most of their time in respect to the whole cell cycle. Mitosis is a very short amount of time in respect to the whole cell cycle. But mitosis is a critical process because this is where it is going to divide and make more cells. Before we get into the steps of division, it's really important to understand that your cells have something inside them, an organelle called the nucleus. And the nucleus holds your DNA. DNA is really important because it's your genetic information. And if you're going to make more cells, you need to have the same DNA in those new cells as you did in your original cells. You want it to be identical, no mistakes. Very important. The problem is you've got a lot of DNA, and we've got to get that DNA into the new cells using mitosis. So there has to be a better way to organize that DNA. Well, what actually happens is that DNA can be organized into these condensed units called chromosomes. Chromosomes are made of DNA and protein. You've probably heard before that humans have 46 chromosomes. That means 46 chromosomes are found in most
That's the number you need to
bodies'
know.
nuclei.
26 chromosomes.
What are nuclei? Well, it's the plural of nucleus. You don't say nucleuses, you say nuclei. Well, in the nuclei, there are 46 chromosomes. Organizing DNA into condensed chromosomes makes it a lot easier to move over when you're making new cells. So if you have 46 chromosomes in a human body cell, you have to duplicate those chromosomes in interphase before mitosis starts.
It's definitely something you need to know. We'll talk about interphase. I will say that that is the same, what she just said is also true for meiosis. Before cellular cells can divide, right, before they can reproduce, they have to duplicate the stuff that's inside of them. And what this video is focusing on is the chromosomes. Obviously, everything has to duplicate, but the chromosomes is what we focus on mostly. But yeah, so whether it's meiosis or mitosis, the cells need to prepare for this division, and that always happens in interphase. And later today, you will learn more about what interphase is.
That basically means you're duplicating your DNA, since chromosomes are made of DNA and protein. You have to do this before mitosis starts because if you're going to make an identical cell that has 46 chromosomes, just like the original, well, it makes sense you have to duplicate the genetic material before splitting. So if you look at our cell cycle video clip, we talk about interphase. That's a stage where most of the time cells are spending their time. They're actually duplicating their DNA during that time. So, ready for the tricky part? Well, because we tend to count chromosomes by the number of centromeres present, when the 46 chromosomes duplicate, we still say there are 46 chromosomes, as the sister chromatids are still attached. And we're
Very important concept. Yes, once those chromosomes are duplicated, you kind of see here, there's one single chromosome in the bottom left. And then you can see here at the right, it's duplicated itself. So now what we technically have are chromatids. We have two sister chromatids. That would be a very important concept. And I'll go ahead and tell you the reason why. Well, one of the reasons why is, again, distinguishing between mitosis and meiosis. When we talk about sister chromatids, that is one of those things that we talk about in meiosis and mitosis. But later, you're going to learn another term that is only for meiosis, and that'll be one of those ways that you can distinguish between meiosis and mitosis. But yeah, definitely need to know what sister chromatids are.
counting by centromeres. So 46 chromosomes here, they replicate in interphase, and you still have 46 chromosomes in this picture, but you went from 46 to 92 chromatids.
Let's be clear here too. I'm glad I'm watching this video first. I don't really care the fact that there's 92 chromatids. That number is almost useless, right? As far as I'm concerned, it is useless. We don't need to. I don't think we need to worry about that. Maybe I need to watch all of their videos, and maybe after seeing them all, it'll all make sense. We go, oh, no wonder they put it that way. But as far as I'm concerned, 92 is not a number you need to know, right? You definitely need to know that we have 46 chromosomes, which breaks down to 23 pairs of chromosomes. But the fact that there's 92 chromatids, not going to be tested on that number. That is a number you do not need to be concerned with.
We have a video explaining that in more depth and how that factors
Burton's
in
here. Good morning,
mitosis.
Burton.
Okay, so now we can get right into mitosis.
Good morning.
I like to tell students to remember PMAT. It's a little acronym that helps you remember. The P is for prophase, the M is for metaphase, the A is for anaphase, and the T is for telophase. So remember PMAT, the stages in order.
And you definitely need to know that. That'll definitely be a test question. It'll ask you to put the stages of mitosis in order, and it is prophase, metaphase, anaphase, telophase. And don't get confused. There will also be a separate test question where you also have to choose, your choice, your multiple choices will be some version of prophase, metaphase, anaphase, telophase in a different order. But the other question is not going to say put them in order. The other question is going to say, in blank, the chromosomes condense. And, then you name the phase, and that would be prophase, just so you know. And then it'll say something like, in blank, the chromatids separate. And you'll learn later that that's anaphase. And then it might say, and in blank, the nuclear envelope reappears, and that would be telophase. And then the next part would say, and in blank, the chromosomes meet in the middle of the cell, and that would be metaphase. So I'll go ahead and tell you now, there will be two questions. They're going to look similar. I'm not trying to trick you. People are going to miss it. So that second question I'm telling you about, where they're not in order, someone's going to guess it in order and then lose points. So make sure you pay attention to the questions.
The very first step is prophase. Prophase because it's the beginning step, the nucleus is still there. And it's going to go away later on, but this is a stage where it's actually still there. The chromosomes are visible. In fact, we say they're condensing, which means they are thickening and visible. The next stage is metaphase.
Speaking of words, now that I think about it, that's a word I've been using for a long time, and I'm going to continue to use it, condense. We say that the chromosomes condense, or they're condensing. That's because usually they're a big stringy mess, but for this process, they need to be nice, compact chromosomes, and I'll tell you why in lecture today. But it just occurred to me, I keep using this word condensing, and in no textbook have I ever read a definition for the word condensing. I guess probably because it just makes sense in context, but just make sure you know when I use the word condensing, that's what I'm talking about, and that'll probably make more sense in the lecture. But again, just so you know, the way they talked about this on this video just made me think of it, like she defining what condensing means, and I've never defined it. I just thought that it made sense in context. But on that note, I'll let you know there will not be a test question where I ask you what condensing means. There'll be plenty of test questions where I use the term condensing to describe something else, but there will be no vocabulary question where I ask you what that means.
M for metaphase, but I also like to remember M for middle because in this stage, the chromosomes line up in the middle of the cell. The nucleus has been disassembled. It's no longer there. So we've got the chromosomes in the middle.
Yes. I don't know if I like this video. Yes, at this point, the nuclear envelope has disintegrated. It has disappeared, so to speak. But the problem is that happens in the previous phase. So prophase is when that happens. So I don't like the way this video just went straight from prophase to metaphase. And then in metaphase, we mentioned the fact that the nuclear envelope is missing, is gone, because that implies that it happened in metaphase, but it didn't. All the stuff that needs to happen to prepare for this process all happens in prophase. So yeah, I'm glad I'm watching these videos now. I don't think I'm gonna share this video with you guys. I don't know yet.
Waiting there. Next, the A is for anaphase. In anaphase, I like to think as the A for away. The chromosomes move away. They are moving to opposite sides of the cell, so they are moving towards the poles of the cell. Now, one thing to point out, these chromosomes, they're not moving by themselves. They actually have something called spindles. These spindles are
Okay, once again, to each their own. And yes, I do talk about the spindles and the centriole, but this, once again, is something that's discussed in prophase. Because obviously, this whole thing about mitosis is moving these little chromosomes around. So before you can start that process, the thing that moves the chromosomes around needs to attach to the chromosomes. And those things are called spindles. And that process actually happens in, you don't need to be quiet or anything, it's all good. That process actually happens in prophase, just like the whole disappearing of the nuclear envelope. So once again, this video will probably be better for, I don't know, middle school or high school, or maybe they didn't ask, maybe their teachers wouldn't ask them, what stage does, what phase are the spindles attached? Because again, that should be a question for what happened in prophase. But yeah, I don't know why they decided to do this video in this order. Oops.
are fibers that help move the chromosomes to the ends. Kind of helps them move along. The last stage of mitosis, think T for telophase. In telophase, the chromosomes are actually at the complete opposite ends, and new nuclei are forming on each side to make these two new cells.
So once again, I think she's leaving out some details that make this a little bit confusing. So to me, this picture is almost identical to what they just showed us for anaphase. So you might be thinking, if this is the first time you've ever seen this or heard of it, you might be thinking, okay, well, what's the difference between telophase and anaphase? This looks exactly the same. So I'll tell you what she didn't say. Anaphase is that process once they first separate. Remember when they were in the middle, in metaphase? Once they first separate and start moving, that is anaphase, right? Anaphase is starts when they first break up, and anaphase keeps going as they move in this direction. And then finally, once they get here to the opposite end, that is when we consider it in telophase. And I can see there's a slight difference in the picture, but I'm not going to say anything. Let's see if the video says anything about it.
The nuclei are starting to surround the chromosomes on both sides. I like to think of the T for 2 because you can really see in this step that the end goal is going to be 2 cells. And in the human body, they're each going to have 46 chromosomes. And again, remember, they are identical. Now, cytokinesis is responsible for the final separation into 2 cells by splitting the cytoplasm, which completes after the PMAT mitosis stages. So why did all this matter? Without understanding cell division, we wouldn't understand how growth and repair happens, because they both require more cells to be made.
And more importantly for you guys, because none of you are biology majors, well, one of you is sort of, the growth part doesn't matter for that person, but this is the kind of stuff that's going to matter for you guys in your regular life as adults when you're dealing with health issues, right? A lot of, as you might imagine, a lot of diseases and drugs and vaccines deal with things like growth and repair. So, if you get a disease and you're doing research on it, trying to figure out, what your treatment options are, so you're not starting from scratch. You are starting from a place of at least a little bit of prior knowledge. Good morning.
Good morning.
Understanding mitosis is also very important for cancer research. Cancer itself is uncontrolled cell growth. So, in other words, uncontrolled mitosis. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
Yeah, anyway, I'm not sure if I'm going to share that video with you guys officially. It could be confusing. In its simplicity, it's too simple, and that makes it confusing.
I just thought that he said, you're right. I'm like, I know. What is it? So much. I'm like, okay. He's like, and look, you can come back up here and see. I was like, okay. He's like,
Reader.
Like, the other thing you were like, so and I'm really just straighten up all of it a lot. It's called a sign. I kind of makes me more awake.
I prefer this to the other room. Plus, it's a little less confusing. I mean, we're used to it now, but on day one, when we first showing up, I'm like, hey, I'm glad you found the auditorium, but by the way, we're also going to meet in a separate place once a week. Only once a week, not 3 times a week. It's at a different time.
But the email confused me. Now it's a 3:05.
Oh, well, I do have horrible handwriting, so.
No, you're good.
All right, start some attendance here. Get that, all right. 4 minutes left to go. All right, let's see. Book is online, Burton. Tape is online. So for those of you who are online early, good job. And I guess you guys too, early, good job. So the next word for attendance, this is not official. These are all extra credit words, extra credit words. So today, these will all be extra credit words. But these are extra credit, if that makes any sense. If not, it doesn't matter. Just send the words in. The next word is scissors, something you would cut paper with. Anyway. Julia is online. Kincaid is online. Fisher is online. Lewis is online. 5 others are online. Let's take a gander here. Gay, Fisher, Lewis, Melton, I already said Crowder. Okay, Crowder's online. This is like triple checking because 1, it's being recorded, so I can always go back to the video to see who's online. 2, I get a report automatically from Google that says this is who logged in, this is when they logged in, this is when they logged out. Actually, and then 3, yeah, you guys will send your words. So anyway, Hannah is online. Listen here. Isabel is online. Durgan is online. Everybody. E. Kincaid, Fisher, Lewis, Melton, Crowder. Yeah, Melton, Julia, Anna, Cook, Durgan, Spade, and Durgan. Okay. Well, got about a minute left. I am going to start really slow today because I don't know how many people are going to show up to the auditorium and maybe miss my sign. I would have made a bigger sign. But. So did you see the sign right when you walked in, or did you kind of walk in like something's up?
I saw the email.
Okay.
It all said 305.
Oh my goodness, it did?
Yeah, that's why I went there. That's why I went there too.
Thanks. I was in such a rush. I was in such a rush to get that out there because I thought time was of the essence. Well, thanks. Well, it's 8 o'clock. I'm going to slowly do attendance.
There's people coming up, but I don't think they know.
The 205 part?
Yeah.
Let me go holler in there really quickly. You guys just relax.
Yeah, but, yeah, 3 o'clock, I just ran upstairs.
Yeah, I'm gonna too. Still for dishes. No. 205, I promise. Oh my God.
I seen it said 305, but I was like, he probably meant 205. So I came here first, and I was like, if he's not in here, I'll just go ahead and go up.
Bro, I checked like every room down the hallway. I'm like, damn.
Yeah, that's the old physics room. One of my least favorite rooms for an undergrad. All right, let's do attendance. We'll get a slow start. So Abigail is here. Melania, Kincaid is online. Andrea is online. Mullins, not here, but possibly online. Lewis is online. Cooper is not here, but possibly online. Hunter is here. Caleb is here. Or online, excuse me. Crowder is online. Reinhardt is not here. Isabel is online. Julia is online. Kennedy, not here. Anna is online. Larissa is here. Madeline is not here. Hughes is here. Galloway is not here. Cook is online. Durgan is online. Tate is online. Burt is online. Kinder is not here. Jones is not here. Canterbury is here in person. Hobson, not here. Well, Tate left the meeting. Anyway, Fisher is online. Did I miss anybody? It's 8:02. I'm trying to go really slow because this last minute change in venue, that could be confusing. So the good news about this is, as far as I know, unless something else happens, this will be the last time we have a change in venue. I have nothing else scheduled where someone else needs to use the auditorium. But don't get me wrong, I prefer it here. Actually, how many of you prefer it here than so some of you, and you guys prefer the auditorium or you just don't care?
It don't matter.
Yeah, it doesn't matter.
Okay. Yeah, I prefer it here, but it is what it is. All right, let's get started slowly. We actually got through the photosynthesis one pretty quickly. So let's see. Again, I'm trying to go slow here. Some announcements and reminders. Welcome to March. This is the last full month of the semester, right? May, we're barely into May and we're done. So this is the last big chunk of the semester. What else to tell you? At the end of this week, we will be in the midterm, in the middle of the semester. What else? Also, at the end of the week, you should have 60 points of independent work. The next exam, I guess technically you could call it the midterm exam, that doesn't hold any more weight than any other exam, should be sometime next week. I'm still working out some details with the people who build the online courses. And I'm trying to get it to where it was like last exam. Otherwise, it'll be on the Google Forms, like the study guides, and I'll have to have you come in. And we know the internet, it can be sketchy here. So we'll see. Keep an eye on your emails for that. What else for announcements and reminders? I also think, in my opinion, it kind of builds the hardness of this course, gets harder and harder and harder until we hit photosynthesis and respiration, kind of levels out in this next section, which is about cellular reproduction and stuff like that, genetics and all that. then it kind of goes down. So we're at the peak, in my opinion, of the hardness, especially with labs. In my opinion, it peaked at the last lab. The photosynthesis respiration lab, in my opinion, is the hardest. It's definitely the one I grade the hardest because it's like the pinnacle. I've been leading you to that lab where like, all right, let's work on hypothesis testing and let's work on looking at your data and seeing if your hypothesis was really correct or not. And I love it. I know this might sound weird, but a lot of you are making a lot of mistakes. But that's good. It's a good learning moment because I can go in and like, your hypothesis was not correct. Look at your data again. So don't feel bad when you get those comments from me. It's a good learning experience. Yeah, you lose points, but you fix the mistake and you get your points back. So yeah, all good news. And like I said in the lab last week or this week, pretty sure that's the last lab that's hands-on. The rest of them are virtual, which again, in my opinion, is easier, but teach their own. I think that's it for announcements. So does anybody have any questions? I would remind you when spring break is, but I don't remember what spring break is. I know it's sometime this month. It's coming.
It starts, yeah,
When?
it starts next Friday.
Next Friday. All right, one more week until spring break. Oh, that would be perfect then. Let me think about this. All right, let's do a vote. Actually, for those of you online, when you send in your attendance words, actually you guys too, do this. You don't need to raise your hand right now. When you send in your attendance words today, also indicate if you would rather have exam 2 this coming week, or if you'd rather have it open over spring break, so then you'd have 2 weeks to complete it. Does that make sense? Like it would be open all of spring break. Not that I expect anybody to do it over spring break, but you can if you want. And then also the following week. So you guys decide. You want, obviously, on one hand, if we do it next week, you're done with it. You don't have to worry about it, and it's fresh in your memory. If we do it the following week, the advantage is you have more time to study for it, and you'll have 2 weeks to do it. So you guys decide. Send those in with your attendance words. And your first word for attendance will be lab, L-A-B, since we're meeting at the lab today instead of a regular auditorium. All right, I've dragged my feet enough. Let's get going on this discussion. So to remind you, Wednesday we finished everything that's going to be on the second exam, right? The second exam is all about energy, right? Why do cells need energy? What particular molecule do they use for energy? What particular molecule do we break down? So the answer to that last one was ATP. What particular molecule do we make, or do we break down to get ATP? The answer is glucose, at least for the exam. You know we break down all kinds of stuff. Where do we get the glucose? Right? The answer is photosynthesis, so on and so forth. This next section is all about cellular reproduction in a way. Another way of putting it, in my opinion, is it's all about chromosomes. So we're going to learn about, in this chapter, we're going to learn about how cells duplicate. So this is called reproduction at the cellular level. And then later we're going to learn, on that note, we're going to learn about heredity. And heredity, when you break it down, it's all about chromosomes. Like which chromosomes of yours came from your mom, which came from your dad. When you produce sperm and eggs, which of those chromosomes are you giving, to a particular sperm or egg? So that's the kind of stuff we're going to talk about. I also will mention this, a good reminder, especially now. This is only the second semester where I've been using this textbook, so I'm still getting familiar with it. It might sound weird to you because you might think biology is biology, and it generally is. But this textbook we're using now is a completely different order than the old textbook. And again, I know that sounds weird, but I've been doing that old lecture, that old PowerPoint, those old PowerPoints since 2016. Now, granted, I do change them a little bit in between every semester. I update them, change this, change that. But I could do that one by memory because I've been doing it for so long, right? This one I'm still getting used to. So that being said, please forgive me if I seem a little bit out of place for this chapter. And on that note, I'm also changing this up a little bit. And you'll see on the PowerPoint slide where I'll say, this is in a different order than your textbook. So I'm still teaching the same stuff that's in your textbook, except I'm doing it in a slightly different order. So there we go. Let's move forward. This picture is not from your textbook. This picture is from your old textbook, but I kept it because I like it. It's like a little guessing game. It's a little bit hard to see in here, but we'll try. Can anybody guess what these are? And I give you a slight hint by process of elimination. I can tell you one thing. It's not a set of boogers. It kind of looks like it. But anybody else know what this is if it's not a set of boogers? Any guesses?
Chromosomes.
Good. They are chromosomes. You're going to get really familiar with chromosomes in this next few chapters. Exam 3 is going to be all about chromosomes. Yes, these are chromosomes. Let's see, 1, 2, 3, 4. I don't remember for sure, but I would assume That is a chromosome abnormality we're looking at, where somebody has an extra chromosome. But I don't know that for sure because I don't see the whole set of chromosomes, which we'll talk about later. Here's one that's a little bit harder because it's hard to see. But can anybody tell what this is? It's really hard to see there.
DNA.
Yeah, it is DNA. And you're going to get really familiar with DNA in this next section, right? This next chapter is going to be a lot about DNA. Because guess what? Chromosomes, they're made up of DNA and other stuff. The important stuff is the DNA. So here's a picture of a lady standing next to a thing of DNA. What in the world? Why do you think they put that together? If you can see, maybe, I don't know if it's easier for you to see. There's like a little arrow pointing from her to the DNA. Any idea what they might be getting out there? Let me fix this while I'm at it. Sorry. That was a good reminder. Anyway, what they're getting at there is that a single strand of DNA is about 6 feet tall. So if you were to take her cells, and stretch out one strand of her DNA, it would probably be taller than she is. That's trivial. It's interesting, but it's trivial. I'll tell you that, and I'll tell you that again later. And I also think it's something mentioned in the lab. But despite how many times it's mentioned in the lab, it's actually quite unimportant. I'm not going to test you on that. Anyway, any idea what this picture is? This is a little bit tougher. In a broad sense, forget about that glowy part, but in a broad sense, what's that a picture of?
X-ray of.
Yeah, some sort of medical imaging, right? X-ray or something. Then how about this glowing thing? Any idea what they're getting at there?
A heart.
That would be a good guess because that's exactly where it should be. And but if this was a different context, you'd probably be correct. But what they're getting at here is cancer. That's a tumor. And this is the chapter where you first start learning about what cancer is. And then in subsequent chapters, this is all going to come back to cancer in a way. Because genetics has something to do with cancer, heredity, all that stuff. Mutations, we'll learn all about it. Anyway, let's move forward again. So again, a lot of this stuff came from the old textbooks and I kept it in. I'm not going to talk about this because I'm not going to test you on it, but it's really interesting, so I kept it on PowerPoint. You can read about it. There's something called parthenogenesis, and it's when females can have babies without having sex, basically. They just clone themselves. It's rare among vertebrates, but there are some species that do it relatively frequently. If you've ever seen Jurassic Park where they talk about, they're all females, like the first Jurassic Park, and then they start making, laying eggs. Like, well, how does that happen? They're all females. This is how, because there's something called parthenogenesis. Of course, they weren't really specific and said something about the frog DNA. And I guess frogs were more known to have parthenogenesis than dinosaurs. But anyway, look it up for independent work if you want. For example, like it says right here we're talking about a shark, but then this slide says it has been documented in about 70 species. So you can look up all kinds of small things about this for independent work. How many? Because exactly how many? Because that says about how many. Plus, this is an older PowerPoint. So you can look that up for it if you want. Which other species do it? How many species of them do it? Is there any kind of relationship? Like, is it mostly sharks? Is it mostly amphibians? Is it mostly reptiles? Anyway, any questions about this slide? All right, this is still all about that. You can read the PowerPoint if you want. All right, so this chapter is broken down into 2 parts. The first part is what the cell reproduction accomplishes, and then finally the cell cycle and mitosis. And in their previous textbook, this used to be all one chapter. This and the next one. The next one's going to be about meiosis. In my opinion, they should be together, but this new textbook breaks it up. First thing you need to know is what reproduction is. So usually when people think of reproduction, they think of 2 things, having sex and having a baby, right? So get that out of your mind. Obviously, that is a type of reproduction, but that's not the type we're talking about here. We will discuss that briefly because what we're talking about is a part of that, but it's not going to be the main discussion we have here. So reproduction, again, like I said, for that first bullet point, may result in the birth of a new organism. Again, like I said, 2 things having sex, they have a baby. But more often, it involves the production of new cells. Right? So that's usually what reproduction is. How many of you have reproduced? I usually get this when no one raises their hand. In all actuality, you should all be raising your hand right now, because not only have you reproduced, but you're reproducing right now. It's this second bullet point right here. More often than not, reproduction is the production of new cells. And you are, as you're sitting here, producing new cells. So you have reproduced, you are reproducing, and you will continue to reproduce. Knock on wood. You know, if you die, you're not going to be. Well, that's a good question for independent work, and I don't even know it. When you die, how long does it take before all your cells start to reproduce, stop reproducing? Probably pretty quickly, because as we know from the last exam, the last unit, without oxygen, cells don't make ATP. Without ATP, they die. Are there some kind of like peripheral cells that can survive on fermentation alone? Anyway, any questions about what reproduction is? All right, the cell undergoes two types of cellular reproduction, which is also known as cell division. I'm not going to test you on that, but just in case you hear me say these words interchangeably, just know they're the same thing, right? Whether I'm talking about cellular reproduction or cellular division, I'm referring to the same thing. But anyway, the one we're going to talk about, the first one, I'm not going to give the name yet, but in this process, we have two daughter cells, right? So we start with one cell, we end with two daughter cells. In this one that we're talking about. I'll give it a name soon. So start with one, end with two. So these two, that looks horrible. But those two cells there, they are genetically identical to each other, right? So this is identical to that, and vice versa. And these things are also identical genetically to the parent cell. That is definitely something you need to know. Actually, I'll go ahead and tell you, even though it's going to be on the slide later. What we're talking about right now, the particular type of cellular reproduction is mitosis. Right, so in mitosis, I'm just going to abbreviate there. In mitosis, again, the product is two new cells that are genetically identical to each other and genetically identical to the parent cell. Any questions about that? It's very important. So a lot of this portion of exam 3 is going to be comparing and contrasting. So remember, like I was saying, in the last exam, or the last stuff we talked about, a lot of that exam is going to be like, at which stage does this happen? At which stage does that happen? We're going to have a little bit of that in this chapter too. But in this chapter, there's also going to be some comparing and contrasting. So I'll be saying, how is mitosis different than meiosis in this way? How are they different in this way? How are they the same? So this is one of the ways that mitosis is different than meiosis. So when we get there, that's one of the things you're going to need to know. How many daughter cells are produced in the other one? And also, are they genetically identical to each other and to their parents? We'll cross that bridge when we get there, but as you can imagine, the answer is it's the opposite of this. Well, we'll get there. Anyway, any questions about this slide? The next word for attendance, because it almost looks like it, water. It's like a stupid water molecule that I drew. With extra oxygens instead of hydrogens, but whatever. Water is the next word for attendance. All right, so before this can happen, before a cell can divide, the chromosomes have to duplicate. And we're going to talk a lot about that. So this is still just a preview. I would ask you if there's any questions about that, but let me explain it first, which comes later. Then I'll take questions. But for now, you do need to know what a chromosome is. And the definition is actually going to get more complicated as we move through this chapter. But for now, when you think of chromosomes, you should just think structures containing DNA. Right? You guys are familiar with DNA already. We learned about it when we learned about biological molecules. Yeah, so for now, when you think of chromosomes, just think structures containing DNA. And I'll also point this out too before we move forward. A lot of stuff happens when cells divide, as you can imagine. It's got to be a pretty complicated situation. But for this level course, this 100 level course, most of what we're going to be concerned with is the chromosomes. There'll be a little bit, but most of the story we're going to tell about the cells dividing is all about the chromosomes. Right? So yes, there's other stuff happening. Yes, your mitochondria have to do something. Your organelles have to do something. And we might scratch the surface of that, but for the most part, we're just going to focus on the chromosomes. Cell division plays lives in organisms. I might ask you this, I might not, I'm not sure, but this is the roles that they play. They replace lost or damaged cells, which is why I said, like I said, you guys right now, as you're sitting here, you are reproducing because there are cells getting repaired or replaced. Also for growth. At one point, when your mom's egg met with your dad's sperm, at that one point in your life, you were one cell, right? How many of you right now are one cell? You're not, right? You're not just one gigantic cell. So when you grew, it's not like that one cell got huge, right? You grew by duplicating your cells. And then finally, reproduction, as most people talk about. So this thing has sex with this thing, they have a baby. That's what that bullet point is right there in this context, right? So yes, more often it's replacing loss of damaged cells or growth, but then yes, sometimes it is used for actual sexual reproduction. Let me put a line through this, because basically, what we're talking about here is this is mitosis above this line, and this one's meiosis. So again, we'll talk about that when we get there. Any questions so far? Okay, here's some pictures from your old textbook, just some examples of what we're talking about. Here's a kidney cell. You can see actually it looks like two cells. It's already duplicated itself. There's one of the nuclei, there's the other nuclei. I mean, they're still together, but you can see that they're kind of split apart right there. You're going to learn about that in this chapter. Here's an embryo. Like I said, at one point you were one cell when you were an embryo or a zygote, and then you doubled yourself. So then you were 2 cells. Then you doubled yourself again, so you were 4 cells. Then you were 8 cells. Then you were 16, 32, and 64, and so on and so forth. That's a great visualization of that. And this is something also, by the way, that we're going to get into, and that stuff is called asexual reproduction. Oops. No, this is all asexual reproduction. Some other examples are single-celled organisms, right? An amoeba, for example. And this is just an example, so you don't need to know it for the exam. But yes, an amoeba is a single-celled organism, and it reproduces via asexual reproduction. Clippings, this is fun for me. I like this. So if you buy like fresh basil, for example, from Kroger, and you still have some left over, you put it in water, it'll sprout roots, and then you can put that into the ground, and now you've got a brand new plant from clippings, and that's because of asexual reproduction. Similarly, again, these are just examples. You don't need to know these exact examples, but can anybody see what this is a picture of right here? How about this? What's this a picture of?
The starfish.
Good, it is a starfish. Not that you need to know that. How about this then? It's also a starfish. So what's happened is if you cut the arm off of a starfish, not only will the starfish grow a new arm, but the arm that was cut off will grow a new starfish. And that is, again, asexual reproduction, right? Just some examples. And if you're into that, you can look up some more examples of asexual reproduction. There's all kinds of animals that do that. Like a lot of lizards, if you try to grab them by the tail, they'll just rip their tail off and they'll grow a new one. Or that's really good examples. Sponge. So if you get this big old sponge and you push it through like a spaghetti strainer, you just have like thousands of pieces. Each one of those thousands of pieces, if you put it back in the water, will go on to become a new sponge. So just stuff to look into for independent work if you want. Any questions about this slide? Just a visualization of some of the stuff we're talking about. Again, talking about mitosis right now. All right. So again, mitosis is a type of it is asexual reproduction. It's how things reproduce asexually. You definitely need to know what asexual reproduction is. There will be questions. We compare and contrast asexual reproduction to sexual reproduction. So again, let's talk about asexual reproduction. In asexual reproduction, there's no sperm or egg. Also, the parents and the offspring have identical genes. Now, I'm not talking about Levi or anything like that. I'm talking about genes with a G. Asexual reproduction is also single-celled, or excuse me, this is also how single-celled organisms reproduce. Mostly, there are some exceptions, and if you want to look into that, what are those exceptions? You might, if you're into it, you can really go down a rabbit hole with that one. Fungi are a very interesting kingdom. Some of them do this. They do reproduce single-celled. Some of them are single-celled, and they do obviously reproduce. But it's not necessarily, it's definitely not asexual sometimes. And it's not the kind of sexual that we're going to learn about. So if you want to, you can dive into that. Like some fungi have more than 2 sexes. Some of them have hundreds of sexes. right? So for humans, and most things that we think of, it's in male or female, not for some fungi. Anyway, moving forward. Again, asexual reproduction. We're talking about multicellular organisms, right? We don't produce new offspring asexually, but we still use asexual reproduction, like I already told you earlier, right? For growth and maintenance. And then there's other multicellular organisms that do use it, like I already talked about in the last slide, to grow new things, right? So a sea star, if it loses an arm, like I said, it'll grow a new arm. Anyway, we've already talked about that. I did that when we had that picture up on the slide. So, any questions so far about asexual reproduction? We'll talk more about it. All right. So again, it's mitosis that's responsible for asexual reproduction. So again, for the exam, you should associate mitosis with asexual reproduction. And even though I'm going to tell you this later, and it's not written on the screen right now, you should associate meiosis with sexual reproduction. So again, mitosis, other than the stuff we just talked about, which was asexual reproduction, it's also used for growth and maintenance of multicellular organisms. So I want to be very clear with that. I think that's one of the bigger mistakes that people make. So when we produce new humans, we need meiosis to produce sperm and eggs. So sometimes people get caught up on that, and they just think, oh yeah, humans do meiosis, and single-celled organisms do mitosis. And it's not like that, right? Single-celled organisms, everything does mitosis. Everything does mitosis. We do mitosis too. Sort of like that thing with photosynthesis and respiration, right? Everybody knows that plants photosynthesize, but then they get so caught up on that fact that they forget that plants also do respiration, right? So they do photosynthesis, they do produce glucose, but they still can't do anything with the glucose just like we can, unless we break it down to make ATP. Anyway, any questions about this slide? All right. Sexual reproduction, which is what we're going to talk about in the next chapter, is the opposite, right? So remember, like I said, asexual reproduction doesn't have sperm or egg. Sexual reproduction definitely has sperm and egg. As a matter of fact, we have a name for that, and you need to know what that name is. Not necessarily because I'm going to ask you what this name is, but because I'm going to use this term a lot. Rather than say sperm and egg, I could just say gamete. And who knows, I'll probably use both terms. I might say sperm and egg, I might say gamete. Just know when I use those terms, I mean the same thing. So the production of these gametes is what meiosis is for, specifically, right? So meiosis produces the sperm and egg. And then after that, we go back to everything. Everything else is back to normal with mitosis, right? The sperm and the egg, they come together. And then at that point, you are a single-celled organism, briefly. And then you start reproducing, right? That one cell duplicates. And then those two cells duplicate. And those 4 cells duplicate. And that process is back to what we were talking about earlier, which is mitosis. So again, meiosis is strictly about producing the egg and the sperm. So any questions about that side? Again, next chapter is going to be all about meiosis. So this is just scratching the surface. So once again, I feel like I'm repeating myself, and I probably am, because I'm still, again, in the transition between the last textbook and this one. But once again, there are 2 kinds of cellular division in the lives of sexually reproducing organisms like us, and trees, and flowers, and cats, and lizards, whatever. The 2 types of cellular division are mitosis for growth and maintenance, and meiosis for reproduction, right? So if we're talking about sexually reproducing organisms, such as ourselves, this is how it works. This is all very important. The one thing that makes this section of the semester harder than the other portion, and again, I think they're about equal as hardness, but this portion is harder than the other in that, where was I getting with that? Sorry. Oh yeah, this is a lot of, you have to understand this before you can understand the next one. So this chapter, you can't really understand meiosis unless you understand mitosis. Then we're going to move past that and start talking about heredity. And you can't really understand heredity unless you understand this. Then we're going to start talking about stuff like evolution. And you really can't understand evolution unless you understand this and heredity. So this is all very important. This is all cumulative. So this is a little bit, this portion is a little bit like math. You know how it is. If you've had a math class, if you didn't understand chapter 2, then there's no way you can do chapter 3 and chapter 4, right? Because it all builds on it. Unfortunately, we are into that portion of biology. Anyway, that's it for the first main bullet point. Pretty simple, right? Any questions about that? All right, the next main bullet point is a lot longer, it's a lot more complicated. We're going to talk about eukaryotic chromosomes. You already know what eukaryotes are. We'll touch back on that here in a second. Then we'll finally talk about duplicating the chromosomes. Then we'll talk about the cell cycle in general. Then we'll finally get into mitosis and cytokinesis, and you'll learn what that is here later in the chapter. And then finally, we'll talk about cancer cells, which is basically cells that are dividing out of control. So let's jump into it. So, in eukaryotic cells, most genes are located on the chromosomes in the nucleus. As far as we're concerned, all the genes are located there. But you can look up for independent work what the exception is. Also, you can look up for independent work. Again, remind you, we're talking about eukaryotes here, right? The more complicated organisms, like humans, or all animals, right? Animals, fungi, plants, protists, these more complicated, larger life forms. Yeah, that's what we're talking about here. As opposed to prokaryotes, which are things like bacteria. So getting back to what I was trying to say, that is another good, decent independent work topic. So everything I'm going to teach you right now has nothing to do with bacteria. So as I'm teaching you all this stuff, you can think to yourself, well, how does this work with bacteria? What do bacteria do here? And you can look into that for a minute. Anyway, it's already right here, sorry. I forgot. I asked the question and I give you the answer. So yes, where are these other genes located? Some of them are found in the mitochondria, some of them are found in the chloroplast. Obviously, we don't have chloroplasts, so we don't have to deal with that because that's only plants and other photosynthetic organisms. But yeah, we do have mitochondria. And I mentioned this before previously in the semester. But to me, this is interesting because your mitochondria DNA only comes from your mother. It's not a mix of your mother and your father. That is your mom's DNA. Because you got her mitochondria. That sperm cell of your dad's didn't have mitochondria in it. So when they met, the only thing you had was your mom's mitochondria. And guess where she got hers from? Her mom. Not her mom and her dad, but her mom. And she got that from her mom, so on and so forth. So that, to me, that's what's fascinating about mitochondrial DNA. You look at your mitochondrial DNA, like if you do 23andMe, that is the same exact, well, other than mutations, it's the same exact genes as your mom's, your grandmom's, your great-grandmom, your great-great-grandmom, all that. All down the line. It's exactly the same, no modification other than some DNA. Excuse me, other than some grief. Mutations. So anyway, look that up for independent work if you find that interesting, look into it deeper. Here we go. Each chromosome contains one long DNA molecule that has thousands of genes. You need to know that. So again, DNA, like we said earlier, it's just a bunch of chromosomes or just a bunch of DNA. So far, that's still the definition. It'll get more complicated later, but right now, when you think of chromosomes, you should think of DNA. It's just one long strand of DNA. Your book says it has thousands of genes. You could look that up. What's the average? You have 23 pairs of chromosomes, and they're all different sizes. So they all have different numbers of genes. So on average, how many genes does a chromosome have? Not that you need to know it, but you could look it up for independent work. And then you could look into other stuff. It doesn't have to be humans. If you like puppies or whatever, look up that for what's the average number of genes for a dog chromosome, so on and so forth. Speaking of which, the number of chromosomes depends on the species. I'm not necessarily going to ask you that, but again, a good independent work topic if you want. What you do need to know is that humans have 23 pairs of chromosomes, right? We have 46 chromosomes, 23 pairs. That is something you definitely need to know for the exam. But the other one's not so important, but slightly interesting, right? So you can look it up if you want. How many chromosomes do bald eagles have? How many chromosomes does watermelon have? Chromosomes does E. coli have? Or whatever it is you want to look up. Oh, excuse me, E. coli is a bad example because it's bacteria. It only has one chromosome. Anyway, then there's another term you need to know. We're going to use it a lot through this section. Genome. You need to know what a genome is. Not necessarily because I'm going to ask you, because I probably won't, but I might. But I'm definitely going to use it a lot. A genome is a cell's complete complement of DNA. So basically, it's all the DNA in a cell, right? That's the genome. And there's all kinds of gnomes that we don't talk about in this class. There's something called a transcriptome, a bunch of other stuff. But anyway, that's what a genome is. Any questions about this slide before we take a look in this bullet point. All right, this picture's from your old textbook. You definitely don't need to know this. If this was a 5th grade science class, I would definitely have this on the exam with this picture, and I would say, how many chromosomes does a giraffe have? And the 5th grader would look and say, oh, a giraffe, that's 30. But I'm not gonna do that. I don't think so. If I did, it would be just for 3 points. Because hopefully that would be really easy, right? You just look at it. But anyway, there it is. It's interesting to me. What I like about this from your old textbook is I think they chose these species on purpose. Because you might think, all right, here's a human, right? And then here's a giraffe. And you might think giraffes are larger, so they have more chromosomes. But they don't, right? And then like this, the largest one here, the largest genome, this one has 102 chromosomes, right? That rat has 102 chromosomes, which is tiny. And then meanwhile, this larger animal, this deer, has only 6. So I don't know, to me, this is all interesting stuff. And again, like I said, decent independent work topic if you want to look into it. Choose some species and look them up. How many chromosomes do they have? Some other things to note too, and I haven't looked into this, I don't know. So if we look at this, we can clearly tell that, for example, this rat has a lot more chromosomes than a dog. But what we don't know is the size of those chromosomes, right? So I'll just draw it out here, try to. Dogs might have 78 chromosomes that are like that size. And maybe that rat might have 102 chromosomes that size. Oops, I guess I should draw 2 of them. Anyway, you don't know. So again, we're looking at the chromosome number, which is one story, but what we can't see from this picture is the actual genome size. Right? How many genes does this rat have? How many genes does this dog have? How many genes does this bison have? So on and so forth. Anyway, again, this slide, while we're doing X's, I'll do another big one. Not important for the exam. I'm not going to ask you those questions. Other than this, humans have 46 chromosomes, right? 23 pairs, 46. Any questions about this slide? The next word for attendance, since it's up here, is going to be koala. Another very interesting independent work topic if you want to look into it. Koalas are some of the dumbest mammals on earth. They're really, really stupid. They will starve to death because if you don't give them eucalyptus leaves on a eucalyptus tree, they won't eat it, they'll starve to death. You give them leaves, but they're too confused if it's not on a tree. Of course, this is what I've heard, and I think I've read it. But I might be wrong. Again, independent work. Look it up, see if I'm right or not. Also, a lot of them are dying from STDs. They're giving each other, I think it's chlamydia is the problem, but they have a big STD problem. Again, interesting topics to look at too if you want. All right, yeah, this one's a little bit information dense, but let's jump into it. So like I said earlier, when you think of chromosomes, you should think of that's the thing that's a DNA strand, that's a strand of DNA. And that is true. You should still think of chromosomes and think that's a strand of DNA. But let's get a little bit more complicated here. Chromosomes are made of something called chromatin. It's not necessarily that I'm going to ask you that, but I'm going to use that word, chromatin. And I'm going to tell you now what chromatin is made of, and that will be important later. Right now it's trivial, but later, knowing what chromatin is made up of will make a difference, because I'm going to tell a story of something that happens to your chromosomes, and without knowing what chromatin is, it wouldn't make any sense. So let this stick in your head here. Chromatin is usually thin fibers, much longer than the nucleus itself. So again, they must be crumpled in there, right? So like I said, if you have a strand of DNA that's 6 feet long, imagine squishing that into a cell so tiny you can't even see it with your naked eyes. So think about how squished they must be. Generally speaking, chromatin is made up of 2 parts, protein and DNA. And again, that will be important later. But for now, just know that chromatin is made up of protein and DNA. Just for your own knowledge, it's about half and half, right? So the same amount of DNA is the same amount of protein. Yet another good independent work topic. What do I mean by that? Same amount of protein versus DNA? Does that mean by weight? Does that mean like if you were to break it up, it'd be like, all right, we've got this, the protein weighs this much and the DNA weighs the same amount? Or does that mean by molecule amount, right? What do I mean by the same amount? You can look it up. If it was important, I won't tell you to look it up. But for now, just know that it's about the same. And that won't be important. The amount won't be important. But definitely the fact that the protein is involved, that will be important later. Why? Well, the proteins organize the chromatin, and they control the activity of the genes. That's not going to be a test question, but that will be important later, especially this bullet point right here. The proteins control the activities of the genes, which will be very important when we talk about how genes work. You already know from the earlier chapter that genes are instructions on how to build a polypeptide. We're going to have a whole chapter of how genes build a polypeptide, and this has a lot to do with it. Anyway, like I said earlier, if we're talking about human DNA, I'm going to put an X to this because I'm not going to ask you this, but it comes up a lot. I don't know why biologists love to talk about this. I personally don't care that much, but a fully extended DNA molecule is over 6 feet long. And if you want to, again, another independent work topic, if you want to do a little bit of basic math, you can look this up. How many cells are there in a human body on average? And then multiply that by 46 chromosomes per cell. And then multiply that by 6 feet per chromosome. And then you can see exactly, not exactly, but about how long your genome is. If you were to take every single one of your cells and break them open and stretch out all your DNA end to end, how long would it be? Of course, you might come up with a number that makes no sense to you, in which case you might want to take that number and compare it to something else. Like it's going to be a really big number. So it's like, what? How far is that? Is it the distance from here to Japan? Is it the distance from here to the space station? Is it the distance from here to the moon? Look it up if you want. Anyway, as the cell prepares to divide, I'm going to put an X to this only because I'm going to talk about this later in more detail. So right now, this is just an introduction. So if you take notes, there's no need to write it down yet. This is the only reason I'm putting an X through it. As the chromosomes coil, or excuse me, as the cells prepare to die, the chromosomes will coil up. We call that condense. I'll talk about that later. It forms the compact structures, like she said earlier. I showed you that booger picture. And she said, no, those are chromosomes, not boogers. And that's what we think of when we see chromosomes. When we think of chromosomes, that's the picture that you should think of. But again, that's when they're all condensed up. That's when they're coiled up and they're condensed. Usually they're not like that. Usually they're just long, squiggly messes that you can't really even see with the naked eye. Anyway, any questions about that side? Long story short, you need to know what chromatin is, and you need to know especially the fact that there's still proteins involved in this, and that'll be important later. So again, there's good pictures of chromosomes. It's a little hard to see with the sliding of this projector. It looks like a bunch of like purple sausages or something, right? That's what chromosomes are. And you can see them there. Here, this is like a big squiggly mess. In this case, it's not the problem with the projector and the light. It really is just a big squiggly, blurry mess because the chromosomes haven't condensed. They're just a big tangly mess. You can't see one chromosome from the other. Here, I can see, yep, that's a chromosome, that's a chromosome, that's a chromosome. So this DNA, we're going to talk about this in a second, but the DNA itself, when we're talking about chromosomes, is packed into an elaborate multi-level system of coiling and folding. Here's some terms you need to know, not because I'm going to ask you, but because I'm going to use them. And again, this stuff I'm teaching you now is going to be more important later, but now is the appropriate time to first teach you about it. And then later, this stuff will come back and it's going to be important. But anyway, histones are the proteins used to package the DNA. So like we said previously, a couple slides ago, chromatin is made up of DNA and proteins, right? So now, not only am I saying it's made up of proteins, I'm giving you some words. Some of these proteins are called histones. And again, I'm going to use those terms, so you need to know what they mean. Then we have something called nucleosomes. So here we have the histones, and then once the DNA is wrapped around those histones, we technically call that a nucleosome. And I'll show you what I mean on the next slide, and then I'll take questions. So there's the double helix, right? The DNA double helix. You can see these little histones, like little protein balls. There's 8 of them. They all come together. The DNA wraps around it. And for each one of those little 8 balls of histones the DNA is wrapped around, that's called a nucleosome. So again, that will be really important later in the semester. Not in this portion. But now is the time to mention it, because now is when I'm first talking about how DNA is really put together. So anyway, any questions about the relationship between DNA, histones, and nucleosomes? Okay. And then there's the bigger picture, right? So it's so interesting. Not only, here's what we were looking at earlier. That's one nucleosome, that's another nucleosome, that's another. So again, it's the DNA wrapped around there. So not only does it do that, but then it coils like this. And then that rope of coils like this. So like I said earlier, this is very elaborate. It's multi-level coiling, right? Very, very compact, very complicated, very ordered. But anyway, any questions about that side? The next word for attendance is written down right here. I'm going to underline it instead of saying it. So if you're online right now and you got your eyes closed, open. For you in the back, can you guys read that? So everybody can read it. And then there's the really big picture. That's everything we've looked at, just all zoomed out. You can see it all in the picture. So let's talk about duplicating chromosomes. We get to the introduction of this. Let you guys marinate on it over the weekend, and then Monday we'll really get into it. But again, before cell division can happen, the chromosomes have to duplicate, right? In other words, another way of saying that is the DNA has to be copied. Right? If you're going to make a perfect copy of your current cell, then obviously you need to copy everything that's in it. You need to copy all your organelles, you need to copy all your DNA. But again, we're going to focus on the DNAs, right? So the DNA is copied. We're going to have a whole chapter on that, or a whole chunk of a chapter that talks about how that happens. But for now, we'll just say it happens. And the result of this is that the chromosome now consists of 2 copies of each original chromosome, and then that's something called sister chromatids. You need to know what a sister chromatid is. That's the term I will definitely use a lot. I guess I should do this on screen. So again, if you had like this one, oh, that looks horrible. This one chromosome, which is doing its thing. Then it duplicates, right? The DNA is copied. And at that point, they're still attached to each other, right? They have this thing called the centromere, right? Centromere, right there in the middle, that dark spot. This drawing is horrible. Those little spikes to the left aren't supposed to be there. Anyway, so there we go. There's a duplicated chromosome. And what we call that is sister chromatids. And that will be an important term. I'm not going to test you on that. But like I said earlier, a lot of this exam is going to be comparing and contrasting meiosis and mitosis. Well, sister chromatids is one of those words we're going to use a lot to compare them. Just so you know, it's because sister chromatids, we talk about that in meiosis and mitosis. Then there's some other version of this I'm going to teach you later that only happens in meiosis. So again, this will be an important term. But that's what a chromosome is, right? I'll show you on this next slide. It'll look better. There's your one chromosome. It duplicates. You can see they're still together, but now technically we call them sister chromatids. So when the cell divides, that means the sister chromatids divide, right? So then, again, they're perfect copies of each other. So they divide, and they go to each side of the cell. So one side gets one perfect copy, one side gets the other. And once they are separated, that moment where they are separated, we then consider them full-fledged chromosomes again. So when they're together, we call them sister chromatids. Once they separate, we call them chromosomes. And the only reason this is important, as far as I'm concerned, is because I'm going to use these terms, and you need to know what they mean when I use them. So I'm not necessarily going to ask you, like, all right, what do you call it when the chromosome duplicates? So what's the result, right? I'm not going to ask you questions specifically about sister chromatids, but I'm going to use this term and this concept a lot. So again, like I said, it duplicates. You've got the one original chromosome, which you can't see here is the centromere. They didn't really draw it. But then it duplicates, so you have sister chromatids. And then those things get separated, and now you have full-blown chromosomes again. The next word for attendance is going to be this. Again, I'm underlining it. You guys see it? All right. A lot of words for attendance today. A lot of extra credit. That is perfect. Yeah, so when we come back on Monday, then we'll talk about the cell cycle. You guys are done at least 45 seconds early. So enjoy your weekend. You're welcome. And thank you guys for your flexibility. I'm sorry that I had my wires crossed with these other people and had to call last minute change. Matter of fact, I give a little extra credit, a little, for your words.
You said we could bring crushed cans?
Oh yeah, while I'm recording, might as well. That's a good question. Yeah, you can bring crush cans for extra credit. It's written somewhere. You might want to find where it's written. It's $5 per bag. But the important thing is it had to be really crushed.
I have a can crusher.
Oh, perfect. That's what I need. Otherwise, the bags are too light and they fly out the back of my truck, and then I'm littering, which kind of goes against the whole self-environment thing. So yeah,
So how does it work? Like
you just bring in a bag
one
once a
bag
week.
equals extra credit, or is it just once a week?
Once a week, you can get like you bring it in, like, oh, there's 5 points of extra credit.
Oh, okay, that's cool.
And then, I mean, technically, you could bring in 2 bags and it's still only 5 points, but whatever.
Do you guys, in this country, do you get money from like, if you recycle the cans and stuff?
Depends on where you go. Some places they do it for free, like in Charleston, you just put it out on your side of the road and they take it and they don't give you money. Here, if you drive it somewhere and go through the work of turning it in, they'll give you money.
I was gonna say, my grandfather gets paid for it. Like pennies though, it's not like
Yeah.
a lot.
Yeah, that bag would probably cost, well, yeah, there's a place in Nitro on Pickens Street, but yeah. You'd probably get, I don't know, 10, 15 cents for a bag, I guess.
Really? In Finland, you can